There's two things that occurred to me that I want to do. Number one is kind of review very quickly, just in words, the idea of the search interface. So there's 3 ways you can do searches on the IEDB. From the home page, you can use the main search interface, which are the 6 panes that are in the middle. And that can do just about everything, but you're not able to access finders that way. So if you want to get at the data tables, you have to click in on search into the results page, which is the second area, the second location of the search panes. And from there, you can access data tables through the finders. The third way is the more advanced, we used to call the advanced search, but that seemed to be a bit of a misnomer. The advanced searches, the specialized searches that Nima showed you, which allow you to get at every single field that we have. Most of the time you don't need those, but there are a few exceptions to the rule, like looking up antibody names and other details that you might want to do. So just as a reminder, there's 3 places where you can get at searches. The simplest one is on the main web page, and then you can click in and get more functionality, and then you can ultimately get at every field if you want to through the specialized searches. So I just wanted to review that quickly. The other thing I wanted to point out, I was talking with Randy in the break, is what I wanted to mention to you is that the data comes in all forms. Some papers are very vague, some papers are very specific. So you notice we have high nodes that are just, for example, a serotype, because often they don't give us the exact allele, they give us a serotype. Even in ligand elution data, we find that sometimes they don't give the exact allele, they give the serotype. So you'll see all different levels of detail because we get all different levels of detail from the papers that we curate. And so the system is designed to handle all levels down to the minute detail and out to the most general things, including just looking up basic responses, T cell versus B cell. All right, so the next section, we're going to run through a couple of example queries, and these are just screenshots walking you through the steps that you would take to perform some basic ones, and we can get more complex as the day goes on. All right. All right, so the first example query we'll do is just one of the most common ones, which is somebody will come in with their epitope of interest and want to look it up in the database. And so in this case, I've picked a peptide that's from influenza as an example. So I cut and pasted it. The fastest way is, as always, cut and paste that there into the text field that's in the epitope search. And note that we are here on the home page, so you can do this from the home page interface. Click search. Let me back up. What this particular example will highlight is performing a sequence search, then performing a homology search of that sequence. So we'll start with doing an exact match, and then we'll do a homology search of different sequence homology. Looking at the results table, and then looking at the summary of the data, and then filtering down and showing you all the details, which you've started to see in some of the other queries that we've run from the questions that have been asked. So I've hit search. And you'll see here on the results page, along the top, you can see the input criteria listed up here. So linear epitope, and then the sequence that we input up there. The epitope tab shows 5 epitopes matching that input sequence from one antigen, obviously. And then 362 assays and 132 references. So this is a very well-studied nucleoprotein epitope from influenza. You can click through the tabs to look at the details. You can see here that the vast majority of the data come from one manifestation of this sequence. By the way, this is a good illustration of how we annotate modifications. So say it was glycosylated or it's a main chain, some kind of main chain modification. It's indicated in this way. So this particular sequence was modified at that residue with this particular post-translational modification. So that's how it would look if you were looking that up, okay? And the reason why these don't have an antigen here, I believe, is just because of the type of modification that took place made it non-natural. All right, so let's see. pushed the advanced button and it went away. I have no idea why. Sorry about that. All right. So now I've clicked open for this particular query. I've now clicked open the assay tab to look at that. So within the assay tab, the 362 assays are subtabs displaying whether or not there were T cell assays, B cell assays, or ligand assays, which would be MHC binding and ligand elution, as I've mentioned earlier. And again, in the parentheses, it tells you the exact number of assays reported for each. So in this case, there's 312 T cell assays, 550 ligand assays, and no B cell assays. I just want to go over the details of this summary. So this table summarizes the assay data, so the context in which this particular epitope was defined, in this case, multiple times in different labs. So this is the ID for the assay. This is, by quick glance, the reference information, the epitope, its position, the protein it came from, and the organism it came from, the host that it was tested in, the immunization procedure, whether it was an administration or administration of a peptide or administration, in this case, of the organism, the assay antigen, which in this set here has been the epitope, and then the MHC restriction, if it's reported, not always reported, but we capture it when it is, and then the assay type and whether or not it was positive or negative. All right, so that's summarized. And again, you can click on each one of these column headers to sort by that. So you can sort by to get all mouse together and all human together. You get the idea. And then I'm clicking open the reference tab to show you that, again, you can click in to see the data from this particular paper. So again, you can drill down from just about every which way in the database. There's the PubMed ID there, the authors, the title, the abstract, and the date, and it's listed in chronological order. Now, the filter feature is a neat feature that actually Bjorn did earlier when he was trying to demonstrate for the non-peptidic Corynebacterium epitope. If you look in the row, along the row next to the epitope, there's this little filter icon. If you click on that and then hit search again, it'll funnel all of the data and it'll reset the data for just that particular epitope. So now it's showing one epitope, one antigen, 358 assays in this case for this particular assay. So then you can just get the data for that particular epitope. You can also filter an antigen. You can also filter an organism. And we can play with that later and show you examples of that. So alternatively, now we're going to look at a homology search. So it's the same sequence that I input, but instead, in this case, we're going to look at homologous structures at a 70% stringency. So and you do this, again, I want to point out, you can do this right from here. You don't have to go back to the home page. You can use what's available here. This will still be there, and you can just quickly click 70% and refine your search in that way. So I'll do that and click search. And this time, you see we get many more hits because the stringency is less because we've said 70% homology. So in this case, you're getting multiple other wild type or natural sequences that are homologous to the one we put in, but we're also seeing analogs. So you see here, this is not reflecting the name of a particular antigen. That's because these are non-natural synthetic analogs of the natural sequence. So say we wanted to look at one, so let's say 733. We'll click on the filter feature to just look at that particular one. And here you'll see the data for that. We'll open up the assay tab so we can see what's there. 2 T cell assays and 2 ligand assays. So you can either click here to drill down, or you can look at the other tab and review the data that's there, probably binding assays, I'm going to guess. But if we click here and drill down, what you'll see is the details, the full details, so all of the fields that we have captured for this particular context, for this particular T cell assay. So I'm going to go through this. The top part is always organized the same. The top part provides information detailing the paper that it came from. So all of this data comes from a paper, and we annotate that here. Title, abstract, links to PubMed, keywords, everything that we could possibly capture that help you get at the information you need. for that particular paper. Then the next box down, or the next set of fields down, details the epitope information. In this case, this is a sequence of the analog we clicked on to look at. It's saying here, this is an epitope analog of this natural sequence here from influenza A. Then if you were to scroll down, there are many fields, so you scroll down, you will then see additional fields. It's hard to actually fit them all on the screen, so I apologize that it's scrunched. But then the details of the immunization procedure. This was a black 6 mouse that was given influenza A. There was restimulation in vitro prior to testing outcome of response. This was a chromium release assay, and it was positive. This is the immunization field that I was telling you about. This is where we provide annotation here for immunization procedure. Then scrolling down, details of the effector cells. Sometimes they are specific. They'll say they were CD8. Sometimes they just say splenocytes. Sometimes they just say PBMCs, but we can capture all of those different details here. And then in this case, the antigen presenting cells were EL4 cells. The restriction was DB. The antigen on the plate was the epitope. So these were mice that were immunized or exposed to the virus and then tested for activity against this analog. Right? And then down here was detailed annotation about the experiment itself and the outcome of the experiment itself. So that's where these kind of details come in. And then also, it tells you what figure, this is something probably we didn't point out earlier. We also capture what figure it came from. So if you go back to the paper, you can quickly find where it is in the paper. And very often when I'm doing this, I'll have this up and I'll have the paper up and I'm looking at details from the paper. And that's the idea, is that you can go from here, quickly find information you need in the database. So you're searching for a needle in a haystack, find the needle, then you can grab the paper and read the details of the paper yourself. So your question about whether or not that non-peptidic, you could quickly look that paper up and find the details of the information, the question you may have had about where that particular antigen came from. So the idea is the interoperability of the database and all the other databases we link to, PubMed being one of them. So any questions about the first example? Yeah.
So the idea of looking at a homologous sequence, how is this search different from if we do a regular NCBI blast alignment of the?
Well, if you do a blast search in NCBI, you'll get a list of all of the proteins that are homologous to that sequence. But you have no link to the papers from which. So this kind of thing gets you, this blast gets you the data in the database, so that the assays specific or related to that sequence. Am I answering your question?
Yeah, so my concern is when we get that hit of all the similar epitopes, we are not restricting by any particular MHC there.
No, unless you want to, but you can.
Yeah, unless I want to. But the lowest level of similarity that you are looking is 70%.
Yes. And
when we know the residues that are going into the pockets of the MHC, we don't need even 70% similarity. Right? Maybe less than 70% is required for the similarity.
Well, it's a starting point. If you already know the antigen that you're working with, I would suggest you could do the search at that level for that particular protein. And look up.
But we cannot go down below 70% here. Right?
Well, if you're searching for sequences similar to that are from a particular protein, but there's different, you can search at the protein level with the restriction that you have and see the epitopes there and compare them to that. So there's even though we have limitations to what stringency we have or lack of stringency in our particular blast search, there's ways of getting at the information that you're interested in getting at.
Yeah, if you're looking for molecular mimicry.
Yeah, you can, yes. I mean, you can get at that because you can do it either looking at the protein level and being very stringent about which MHC restriction that you're applying to that particular protein, and then download that in Excel and scrutinize it that way. And there's other but we can talk about that afterwards, and I can show you ways of doing that. But yeah, good questions. All right. So the second example is going to be looking up T-cell epitopes that have been associated with treatment of allergy. All right. So here, because we're looking up a particular assay type, we're going to go into the results page to access the search panes that have the finder. So in this case, we're interested in the assay finder down here. So I'm going to scroll down here to the assay finder, and I'm just going to start typing treatment. And what will come up in the autocomplete are the terms that are associated with that. So in this case, decreased disease symptoms after treatment, and that will, I will select that one, and then I will scroll down to the disease pane and select just at the high level allergy. Of course, you could come down here and type in a particular type of allergy, but just for demonstration purposes, I'm just clicking allergic disease in general, and then I'm going to click search. So here you see the results of that. 111 epitopes have been associated with the treatment of allergy. So this is all hosts, because I didn't discriminate. And if you click on the assays tab to take a look at all of these, what you'll see is a tremendous variety of different allergens, dust mite, bee sting, grass, fungus, so on and so forth. They are listed here, and right next to them, the organism from which they came. So you see house dust mite, cat, mouse, trees, tree pollens, and other, here's fungus right there. A couple of things about this. I want to remind you, you can funnel and just look at any particular allergen. You can funnel and look at any particular organism that generates allergens. You can also map this to the Immunome browser, and you can look by relative abundance. Here on the Antigen tab, once you open that, you can see that this particular DERP1 has been studied quite heavily, multiple epitopes, multiple assays from a lot of references. And again, you can sort on these columns here to look at ones that have more or less reactivity. Then if you...
Sorry, could you go back to the slide before that?
Sure, yeah.
Before that one?
Back again?
Yeah.
Yep.
Sorry, the criteria you use for the allergy search.
Oh, sure. So just the type of assay, treatment assay, and just higher category allergy.
You focused on cardiolipin.
I'm sorry?
Yeah, so I just noticed that cardiolipin is that the compound you're targeting, or if you look further up in that table, you've got a cardiolipin.
Oh, okay. So that's a good question. This is actually really important to point out. When you start on the home page and come into the results page, and you hit search and you input no criteria and hit search, it's bringing you into the results page, but it's also running a query at the same time. It's querying everything. So what comes up, that just happens to be showing right here. This is reflecting, if you notice the number here, that's the total number of epitopes.
Okay.
So that's a good question, though, because that can confuse people. There's things that we're used to seeing all the time, and we don't think a second thing of it. But it's important to, yes, thank you for bringing that up. That's the only reason why that's there. All right, so on that, so for the treatment assay query, here we're on the assay tab, and again, this is a summary of each record represents a different assay that's run, the lab that it came from or the paper that it came from, the epitope, the host, immunization, the antigen, the relationship of the antigen to the epitope. In this case, it was the antigen that the epitope came from. This is a little bit of jargon on our part, but just so you understand what we mean. And then the assay type. And then here's a good example of where sometimes you get restriction and sometimes you don't, defined in the assay. And again, you can sort on these columns, so you can look for, if you're just interested in human data, here you can quickly look for it there. You can also go back and refine your query and just look for human data that way. So there's lots of different ways to get at the things that you're interested in.